Thank you, Neta. Our next speaker is Professor Eli Pikarsky. It's a great pleasure to invite you to come to the stage. Eli is a professor at the Lutemberg Center for Immunology and Cancer Research at the Hebrew University Hadassah Medical School and the chairman of the Department of Pathology at the Hadassah Hebrew University Medical Center in Jerusalem. He is also the chairman of the research committee of the Israel Cancer Association and is an active surgical pathologist. What is a surgical pathologist? Stam patholog. Involved in clinical work and runs a research laboratory where he studies tissue aspects of cancer development in mouse models. Thank you.
So, thank you, Ronit, for inviting me. I apologize for being late this morning. Apparently, my ability to drive outside the city declined faster than my COVID antibodies. Really, it was a traumatic experience after 2 years of staying in my small town of Jerusalem. I'm always pale. I'm frightened when I stand here. So thank you for inviting me, and it's really a huge pleasure to be in an audience with real people in 3 dimensions. And what we're trying to do is to study immunology in 3 dimensions. And when you talk about the immune system, you have to remember that there is this balance when you talk about the immune system in patients. And there is the immune system is a protective system. It developed along an evolution to protect us. So its main function is protective immunity. But unfortunately, in many diseases, actually in every chronic disease, including cancer, there is immunopathology where the immune system plays pathological roles by promoting disease instead of fending it off. And we heard many examples for that, and also examples on how to try to re-educate the immune system, like Cyril Cohen is doing. So this balance between immunopathology and protective immunity is something that is very important to understand, and this is what we're trying to do. And we're doing it mostly in the context of liver cancer. And liver cancer is one of the deadliest cancers, 2nd or 3rd leading cause of cancer mortality worldwide. When we talk specifically about hepatocellular carcinoma, it's either 3rd or 4th. But it's a major cancer. The prognosis is terrible. We have very few successful treatments. Immunotherapy is now changing it. Interestingly, only when you combine it with VEGF inhibitors. We don't really know why. Lots of theories. I get to write reviews on this, but no real data to support, so you can write whatever you want. And what's interesting about HCC to me is that it, in most cases, I would say more than 90% of the cases, probably more than that, it develops on the background of a chronic inflammatory process that was taking place before cancer developed, in a way disposing predisposing to the development of cancer. And I can say today there is sufficient evidence to say now causing cancer in these organs, likely by promoting its development. So what does the immune system do in cancer? This is it's a review I wrote together with Matthias Heichenwälder, where we tried to simplify what the immune system does in liver cancer specifically. And what you see here is that there are many cell types, and for the red arrows are bad, the green arrows are good. And what you can see, I think, very nicely, first of all, that it's very difficult, that it is a big picture, probably meaning that we really don't understand it well. And we didn't learn a lot since. But the other thing is that every cell can do both immunopathology and protective immunity. And this is something, I think the first person to note this for macrophages was Alberto Mantovani, and he coined this idea of M1 and M2 macrophages, like good ones and bad ones. And today we think about the spectrum and multiple phenotypes which a macrophage can assume. And what this picture shows you is that in the liver and in any other organ that you want, this notion that was originally developed for macrophages is important for any other immune cell that you would want to think of. And now I want you to do something that maybe in the last 2 years we could do more. Use your imagination and think about an immune reaction. Think about an immune response to something and think about the organ, right? As Ronit said, I'm a pathologist. I like to think about diseases in the context of an organ. And so imagine the immune response in your head. And you're probably thinking of something like this, a swarm of cells that are going into the tissue, trying to do something. And I just told you that they're confused. They don't know if they should be good or bad, but they're all coming in and they will decide eventually what they're doing. But the immune system sometimes doesn't look like this, but rather it looks like this. It forms these very organized structures. And this is something that we are now studying for maybe the last 8 years or so. And these are called tertiary lymphoid structures. These are microorgans that developed in chronically inflamed organs. They are composed of multiple adaptive immune cells, B cells, T cells, also some innate immune cells. In my mind, every cell in our body belongs to the innate immune system. And they look like small lymph nodes. They're not lymph nodes because the liver does not have any lymph nodes. They don't develop, lymph nodes don't develop within the liver tissue in embryogenesis, and lymph nodes do not form after you're born. So these are these tertiary lymphoid structures, and they're supposed to be these frontal command posts of the immune system, where the adaptive immune system can generate adaptive immune responses, both of T cells and B cells, that are specific to something that is occurring in the tissue. It's a very smart way to organize your response. And when you think about them in cancer, so Jim Moulet was the first to note in recent years, but this was also described by Rudolf Virchow 150 years ago. The presence of these tertiary lymphoid structures in cancer is usually associated with a good prognosis, probably implying that this is a place where T cells that are active against the tumor, and today we also think about B cells as important players in anti-cancer immunity, are formed. So these TLSs are thought to be good. And I want to begin to convince you today that just like cells can have different flavors, pro and anti-tumorigenic, so these structures, these tertiary lymphatic structures, can be either pro or anti-tumorigenic, and they can do it in multiple ways, okay? So we started this project in collaboration with Eugene Hoshida. He was then at Mount Sinai in New York. And we looked what does the presence of chronic inflammation in the liver of these tertiary lymphatic structures, which I will call from now on TLSs, what is their implication for prognosis? And this was a big surprise, because this was the first time the TLSs were shown to have a bad prognosis in cancer. This is actually not true for TLSs which are found in the cancer. Julien Calderero in France, after seeing this, looked at intra-tumoral TLSs in HCC, and they signify a good prognosis, just like in any other tumors. But then after this was published, others have shown in many other cancers, including also in the liver, in cholangiocarcinoma, very recently, the TLSs in the liver parenchyma can be associated with a bad prognosis. So there are good TLSs and bad TLSs. So what makes these hepatic TLSs bad in this case? And so what we do in order to try to answer these questions, hopefully in a human relevant way, is to develop mouse models. And here, not meaning to do so, but we developed a mouse model that develops multiple TLSs in the liver. We is Shlomi Finkin when he was at the lab. And you can see very nicely that these mice develop more and more TLSs with time. And this is associated with the development of tumors. So more TLSs, more tumors, there is association which is reminiscent of what is seen in humans, and we analyze these TLSs to see that they're also histologically and also from the molecular expression pattern of different cytokines. They're very similar between the mouse and the human. And what was interesting, and I think this was to me where it really became interesting, Shlomi detected the tumor initiating cells, the smallest tumors in the livers of these mice actually developed within the TLSs. So the TLSs became micro niches that supported the growth of the tumor cells, and then we did some 3D analysis. This was done together with Datya Nguyen and Matthias Heichenwälder in Heidelberg. And you see that these TLSs, the tumor cells here in green, they begin to form within the TLS, and then they start budding out of it. So are they forming where they formed, and then the TLS developed around it in trying to kill it, and then they're trying to run away? Or was it a micro niche that supported their growth? And then after they are nourished and they leave their baby period, they can grow and become independent tumors. And to answer this, Shlomi, in several ways, both genetically and pharmacologically and using antibody ablation, ablated those TLSs, and the answer was very clear. There was a marked reduction in the number of tumors. And then I won't go into the data. He identified a cytokine, a lymphotoxin beta, as a proof of concept of a cytokine which is produced specifically in this niche and was necessary for the growth of these tumors. So these TLSs are bad because they provide cytokines which promote the growth of tumors until they gain independence. And we're now trying to understand what independence means, at least in this context. And this is the model. You see that this is the cytokine, and then they grow, and then they go out, and so on. So then we started to ask, okay, so how are they formed? What generates these TLSs in the liver? Is it similar to what happens in other organs? And this is what Einat Cinnamon and Ella Nazirov in the lab are doing. And so very little is known about how TLSs are formed. Basically, it's a copy-paste from what we know about how lymph nodes are formed in embryogenesis. And one cell type that is very important for the formation of these structures are innate lymphoid cells, which kind of, when they appear in a region, in an organ, they can summon B cells and T cells to form these structures. So what Einat did is she just bred our mice with mice which don't have these innate lymphoid cells. And we were expecting to see fewer tumors, fewer TLSs, and it was from the first mouse that Einat analyzed, it was very clear that there is a huge increase in the number of TLSs. So not only aren't these LTI cells necessary for the formation of these TLSs, in this case, in fact, their presence inhibits the formation of the TLSs, or in a way prevents them from forming their function, thus inducing a reactive increase in their numbers. But what about tumors? Are these bad TLSs or good TLSs? And so this you've already seen, but if you look at the numbers of tumors, and even more so if you compare the numbers of the ratio between TLSs and tumors, then there is a marked decrease in the numbers of tumors. So there are more TLSs, but now they know how to do their job. So that was good because now we can compare the TLSs in these mice and these mice, and we used I think I have like 4 more minutes on it, Nahon? Okay, so we use all these, what Matthias called fancy schmancy methods, CyTOF and single-cell RNA sequencing. And these are it's very sophisticated, but we are not bioinformaticians, so maybe we cannot learn a lot from this. But there was something that was very clear from both methods, and that is there is, I will run through this, there is a marked reduction in the, or when you deplete the LTI cells, you have more CD8 cells, and the CD8 cells are more aggressive in a way. They express higher levels of granzyme B and granzyme A and perforin, and they're also less numbers of what we think are dysfunctional T cells. Now here, there is a problem. We cannot isolate these cells and test their ability to inhibit the growth of tumor cells. Everything we're doing in vivo. So what we did in order to somehow characterize the T cell function is to measure the proliferation capacity of the T cells in vivo. So we look for CD8 positive, Cas67 positive, PD1 positive cells, and there is a huge difference. So we think that they are less active because of this. So somehow the LTI cells inhibit the activity of the T cells. So to summarize and leave you time for questions, if you have any, TLSs, just like specific immune cell types, can be good or bad. And when you look histologically, you cannot tell. There is no histological difference between them. Histology does have some limitations, I must admit. I didn't show you all the data about specific cell subtypes. That's the work that Ella is doing by depleting CD4 cells and B cells. And it is when a TLS loses, what we think today is that when the TLS loses its ability to kill tumors by developing high numbers of dysfunctional CD8 T cells, then it can manifest its pro-tumorigenic function as a classical chronically inflamed niche, which many people, including Neta and many others, have shown support the growth of tumors. I want to thank the people that are doing the work. This is Einat Cinnamon. This is Ella Ilan, who runs the lab. And this is in the Israeli Museum in Jerusalem. It's still there. You can come and visit. It's a very beautiful place. And thanks to many collaborators, Yinon, Matthias, Datyan from his lab. We're now trying to see if all this also occurs in melanoma together with Michal and Liat, and the single cell analysis is done in collaboration with Oren Parnas. A lot of funding for all these expensive experiments, and Shlomi, that is now, that was a student in the lab and started all this project, which I find very fascinating, and is now in the Rockefeller University finishing his postdoc. Thank you.
Fantastic talk, Eli. Thank you. Questions? Yes. So many hands.
Eli, fantastic talk, as Ronit said.
Well, Cyril, you're a real immunologist. I'm not, I'm afraid now.
Okay. Well, exactly. And I was in the surgery branch in the States, so I kind of balanced that. Quick question. Have you looked at, or did you find a difference in chemokines and chemokine signaling between bad TLS, TLS2 and TLS1 or something like that?
Okay, so we don't call them TLS 2 and 1, but that's a good idea. It should be 2 and 1 if you want to recapitulate the M1 and 2. And yeah, so there is a difference, for instance, You do these RNA signals. I don't remember, but there are differences. We tried doing this. Nothing very nice came out of it. We are concentrating now on a specific cytokine, CCL20. No, we didn't. We found CCL. We looked for cytokines which are expressed by the epithelial cells, simply because we wanted to identify the epithelial immune crosstalk. And the only one that we really found was differentially expressed was the CCL20 by the epithelial cells, which is known to be expressed by liver cells, so maybe not a surprise. But it's functional because when you knock out CCL6, which is the receptor, you see a marked decline in tumors.
Great talk, as usual. Do you know, I'm sure you do, how general is this phenomenon of tertiary lymph nodes? Is it something that you see only in specific cancer or specific organs? So is it organ-specific or cancer-specific or just a general phenomenon?
It's extremely common. In fact, there is no chronic inflammatory disease where you look at a slide where you won't find one. Maybe you have to look for them. In cancer, they're mostly seen in tumors with high mutational burden. For instance, when you see a colon cancer or endometrial cancer, and today any cancer, with high numbers of TLSs, you should suspect that it is mismatch repair deficient and check it. Today we check automatically for every tumor because of the response to immunotherapy, but yes. The bad ones, we cannot identify them yet very efficiently, so we don't know how common they are. They have been observed by others in breast, in glioma, in transitional cell carcinoma. But I suspect that because the only way to identify them today is by prognosis, is that they're more common, we just do not have biomarkers for them. And there is no good antibodies that can say if a specific T cell is functional or dysfunctional. So when we have those, maybe we'll know more.
Now, yes. Maybe you already partially respond, but I was wondering if now we all asking the question, what is the difference or how can we differentiate between the responders and non-responders to immunotherapy or immune checkpoint? Is there any correlation, or has anyone looked at the role or how the tertiary lymphoid structures change, and if they can predict, response to immunotherapy and how different they are, right?
Yeah, there were 3 back-to-back papers in Nature, unfortunately not by our group, showing that it's, you should have asked me this 2 years ago. Maybe we would have this paper together, yeah.
Great talk. Just maybe I missed it. Are the CD8 T cells in those TLSs, are they antigen specific? And do they need to be exhausted or dysfunctional in order to initiate tumor growth, or this comes later on?
So I don't know if they're antigen specific. I'm constantly asked this question, and I don't have, embarrassingly, I don't have an answer. I'm not sure I know how to answer this. Okay. After I look at the distribution of your CAR T cells, you will explain how I should do that. Okay. And I think, yeah, they first need to be dysfunctional because, if they do their job and kill the tumor cell, then they can't help it grow again. The one thing in biology that is really final is death. I'm a pathologist.
I'm curious about the part that you didn't have time to speak about, the CD4 and B cell depletion. I'm wondering how it affects memory, maybe the germinal center level, if you looked at the neutralizing antibody formation and so on.
Yeah, so we didn't look about the antibody formation, but my hypothesis, and Einat and Ella both do not agree with me, so I'm probably wrong, is that one important role of the immune cells here is to induce metaplasia. That is, that hepatocytes differentiate into cholangiocytes, and somehow these cells are important for this. And so we're doing B cell depletion and T cell depletion and co-depletion. And the problem is, you know, when you deplete one immune cell type, annoyingly the others increase in number. So you have to try to understand whether the effect that you're seeing is because B cells are missing or because you have more CD8 T cells. So that's now we have to do all these experiments in conjunction, laborious, very expensive, but doable, and we'll soon have results.
Thank you.
Yeah.